Good afternoon, classmates, and good afternoon also to our dear professor, Mumtama Abamo. So, my topic is about cell junctions and cell adhesion. By the way, I am Kathleen Ramos Palmero. Let's start with the question, what is cell junctions? So, cell junctions is a specialized region of connection between two cells or between a cell and the cellular matrix, and they are particularly abundant in the epithelia. So, cell junctions is best visualized using either conventional or freeze fracture electron microscopy, which function to reveal the interacting plasma membranes, which is highly specialized in this region. So, by the way, freeze fracture electron microscopy is a technique for studying membrane structure in which the membrane of a frozen cell is fractured along the interior of the bilayer, separating it into two monolayers. Cell junctions can be classified into three functional groups. We have the first one, the occluding junctions, which function to seal cells together in an epithelium in a way that prevents even small molecules from leaking from one side of the sheet to the other. And the next one is the anchoring junctions, which functions as which function to attach cells mechanically to their neighbors or to the extracellular matrix. And lastly, we have the communicating junctions, which function to mediate the passage of chemical or electrical signals from one interacting cell to its partner. So this table shows us the major kinds of intercellular junctions within each group. So we have the three functional classification of cell junctions: the occluding junctions, the anchoring junctions, and the communicating junctions. Under the occluding junctions, we have tight junctions and septate junctions. In the anchoring junctions, we have the two attachment sites, the actin filament and the intermediate filament, in which in the actin filament, we have cell-to-cell junctions or the adherence junctions. The second one is the cell matrix junctions or the focal adhesions. And in the intermediate filaments, we have the cell-to-cell junctions, or the other term is desmosomes. And the second one is the cell matrix junctions, and the other term is hemidesmosomes. And lastly, we have communicating junctions. We have the gap junctions, the chemical synapses in the plasmodesmata, which is for plants only. Occluding junctions form a selective permeability barrier across epithelial cell sheets. So, all epithelia have at least one important functions in common. They serve as a selective permeability barriers, separating fluids on the other side that has different chemical composition. This function requires that the adjacent cells be sealed together by occluding junctions. Also, tight junctions have this barrier role in vertebrates, as we illustrate by considering the epithelium of the mammalian small intestine or gut. So, in this figure below, the epithelial cells lining the small intestine, which forms a barrier that keeps the gut contents in the gut cavity or the lumen. At the same time, however, the cells must transport selected nutrients across the epithelium from the lumen into the extracellular fluid that permeates the connective tissue on the other side. So this long tube-like organ, which is red in coloration, that is the epithelial tissue or the epithelial cells, which forms the epithelial tissue. And the green one is the connective tissue, and the yellow one is the muscle tissue. Each tissue is an organized assembly of cells which is held together by cell-to-cell adhesions, extracellular matrix, or both. So from there, these nutrients diffuse into the small blood vessels to provide nourishment to the organism. This transcellular transport depends on two sets of membrane-bound membrane transport proteins. One set is confined to the apical surface of the epithelial cell, which is in the figure 19.2. The apical surface on the upper part and the basolateral surface, which is located in the lower left part. This apical surface of the epithelial cell is actively transporting selected molecules into the cell from the gut. And this basolateral surface of the cell allows the same molecules to leave the cell by facilitated diffusion into the extracellular fluid on the other side of the epithelium. So, to maintain this directional transport, the apical set of transport proteins must not be allowed to migrate to the basolateral surface of the cell, and the basolateral set must not be allowed to migrate to the apical surface. Furthermore, the spaces between epithelial cells must be tightly sealed so that the transported cells or molecules cannot diffuse back into the gut lumen throughout these spaces. So, yeah, as we can see here, the tight junctions is the one who divides the apical surface in the basolateral surface. Its division is very important since it functions to avoid mixing of the molecules of this of the two regions. So the roles of tight junctions between epithelial cells. The tight junctions between epithelial cells are thought to have both of these roles. First, they function as barriers to the diffusion of some membrane proteins and lipids between apical and basolateral domains of the plasma membrane. Mixing of such proteins and lipids occurs if tight junctions are disrupted, for example, by removing the extracellular calcium ions that is required for tight junctions. Second, tight junctions seal neighboring cells together so that if a low molecular weight tracer is added to one side of an epithelium, it will generally not pass beyond the tight junctions. Although all tight junctions are impermeable to macromolecules, their permeability to small molecules varies greatly in different epithelia. Tight junctions in the epithelium lining in the small intestines, for example, are 10,000 times more permeable to inorganic ions such as sodium ions than the tight junctions in the epithelium lining the urinary bladder. These differences reflects differences in tight junction proteins that form the junctions. So, the roles of tight junctions between epithelial cells pa rin. So, epithelial cells can transiently alter their tight junctions to permit an increased flow of solutes and water through breaches in the junctional barriers. Such paracellular transport is especially important in the absorption of amino acids in monosaccharides from the lumen of the intestine, where their concentration can increase enough after a meal to drive acid transport in the desired direction. So, when tight junctions are visualized by freeze fracture electron microscopy, they seem to be composed of a branching network of sealing strands that completely encircles the apical end of each cell in the epithelial sheath. So let's look at this figure 19.4. which is the structure of a tight junction between epithelial cells of the small intestine. So in this figure A, we have here a junctions that is shown schematically, and in the figure B, we have here a first fracture electron micrograph. In the figure C, we have a conventional electron micrograph. So note that the cells are oriented with their apical ends down. In B, the plane of micrograph is parallel to the plane of the membrane, and the tight junction appears as a belt-like band of branching sealing strands that encircle each cell in the epithelium. The sealing strands are seen as ridges of the intramembrane particles in the cytoplasmic fracture base of the membrane. And in C, the junction is seen in cross-section as a series of focal connections between the outer leaflets of the two interacting plasma membranes, each connection corresponding to a sealing strand in cross-section. So each tight junction sealing strand is composed of a long row of transmembrane adhesion proteins embedded in each of the two interacting plasma membranes. The extracellular domains of these proteins join directly to one another to occlude the intercellular space. The major transmembrane proteins in a tight junctions are the claudins, or which are essential for tight junction formation, and function and differ in different tight junctions. A specific claudin found in kidney epithelial cells, for example, is required for magnesium to be resorbed from the urine into the blood. A mutation in the gene encoding this claudin results in excessive loss of magnesium ion in the urine. A second major transmembrane protein in tight junctions is occludin, the function of which is uncertain, but the claudins and occludins associate with the intracellular peripheral membrane proteins called the ZO proteins or the zooproteins, which is also known as zonola occludins, which anchor the strands to the actin cytoskeleton. So, in addition to claudins, occludins, and ZO proteins, several other proteins can be found associated with tight junctions. This includes some that regulate epithelial cell polarity and others that help guide the delivery of components to the appropriate domain of the plasma membrane. Thus, the tight junctions may serve as a regulatory center to help in coordinating multiple cell processes. So in invertebrates, septate junctions are the main occluding junction. More regular in structure than a tight junction, they likewise form a continuous band around each epithelial cell. But their morphology is distinct because the interacting plasma membranes are joined by proteins that are arranged in a parallel rows with regular periodicity. So a protein called discs large, which is required for the formation of septate junctions in Drosophila, is structurally related to the ZO proteins found in the vertebrate tight junctions. Mutant flies that are deficient in this protein not only lack septate junctions but also develop epithelial tumors. This observation suggests that a normal regulation of cell proliferation in epithelial tissues may depend in part on intracellular signals that emanate from occluding junctions. So, this figure of a septate junction. It shows here in a conventional electron micrograph between two epithelial cells in mollusks. So the interacting plasma membranes seen in cross-section are connected by a parallel rows of junctional proteins. The rows which have irregular periodicity are seen as dense bars or septa. Anchoring junctions connect the cytoskeleton of a cell either to the cytoskeleton of its neighbors or to the extracellular matrix. So the lipid bilayer is flimsy and cannot by itself transmit large forces from cell to cell or from cell to extracellular matrix. Anchoring junctions solve the problem by forming a strong membrane spanning structure that is tethered inside the cell to the tension-bearing filaments of the cytoskeleton. So anchoring junctions has two main classes of proteins. We have the intracellular anchor proteins, which forms a distinct plaque on the cytoplasmic face of the plasma membrane and connect the junctional complexes to either actin filaments or intermediate filaments. The next one is the transmembrane adhesion proteins, which has a cytoplasmic tail that binds to one or more intracellular anchor proteins and an extracellular domain that interacts with either the extracellular matrix or the extracellular domains of specific transmembrane adhesion proteins on another cell. In addition to anchor proteins and adhesion proteins, many anchoring junctions contain intracellular signaling proteins that enable the junctions to signal to the cell interior. So, anchoring junctions occur in two functionally different forms. We have the adherence junctions and desmosomes hold cells together and are formed by transmembrane adhesion proteins that belongs to the cadherin family. Cadherin is a member of a family of proteins that mediates calcium-dependent cell-to-cell adhesion in animal tissues. And the second one is the focal adhesions and hemidesmosomes bind cells to the extracellular matrix and are formed by transmembrane adhesion proteins of the integrin family. So integrin is a member of a large family of transmembrane proteins involved in the adhesion of cells to the extracellular matrix and to each other. On the intracellular side of the membrane, adherence junctions and focal adhesions serve as connection sites for actin filaments, while desmosomes and hemidesmosomes serve as connection sites for intermediate filaments. Adherence junctions connect bundles of actin filaments from cell to cell. So, adherence junctions occur in various forms. In many non-epithelial tissues, they take the form of a small punctate or streak-like attachments that indirectly connect the cortical actin filaments beneath the plasma membranes of two interacting cells. But the prototypical examples of adherence junctions occur in epithelia, where they often form a continuous adhesion belt or the zonular adherens just below the tight junctions, encircling each of the interacting cells in the sheath. The adhesion belts are directly opposed in adjacent epithelial cells, with the interacting plasma membranes held together by the cadherins that serve here as the transmembrane adhesion proteins. So within each cell, a contractile bundle of actin filaments lies adjacent to the adhesion belt, oriented parallel to the plasma membrane. The actin is attached to this membrane through a set of intracellular anchor proteins, including the catenins, the vinculin, and the actinin, which we consider later. The actin bundles are thus linked via the cadherins and the anchor proteins into an extensive transcellular network. This network can contract with the help of the myosin motor proteins, and it is thought to help in mediating a fundamental process in animal morphogenesis. So desmosomes connect intermediate filaments from cell to cell. So desmosomes are button-like points of intercellular contact that rivet cells together. So inside the cell, desmosomes serve as an anchoring sites for rope-like intermediate filaments which form a structural framework of great tensile strength. The particular type of intermediate filaments attached to the desmosomes depends on the cell type. They are the keratin filaments in most epithelial cells, for example, and the desmine filaments in heart muscle cells. The junction has a dense cytoplasmic plaque composed of a complex of intracellular anchor proteins. The plakoglobin and desmoplakin are responsible for connecting the cytoskeleton to the transmembrane adhesion proteins. So, these adhesion proteins, desmoglin and desmocolin, like those at the adherence junction, belong to the cadherin family. So, they interact through their extracellular domains to hold the adjacent plasma membranes together. So, in this figure, in letter A, we have here an electron micrograph of 3 desmosomes between 2 epithelial cells in the intestine of a rat. In the electron micrograph of letter B, it shows here a single desmosome between 2 epidermal cells in a developing, showing clearly the attachment of intermediate filaments. And in letter C, it shows here the structural components of a desmosome. So in letter D, it shows some of the molecular components of a desmosome. Desmoglin and desmocolin are members of the cadherin family of adhesion proteins. Their cytoplasmic tails bind plakoglobin, which in turn binds to desmoplakin. Desmoplakin also binds to the sites of intermediate filaments, thereby tying the desmosome to these filaments. So, anchoring junctions formed by integrins bind cells to the extracellular matrix, focal adhesions, and hemidesmosomes. Some anchoring junctions bind cells to the extracellular matrix rather than to other cells. The transmembrane adhesion proteins in these cell matrix junctions are integrins. It is a large family of proteins distinct from cadherins. Focal adhesions enable cells to get a hold in the extracellular matrix through integrins that link intracellularly to actin filaments. In this way, muscle cells, for example, attach to their tendons at myotendinous junctions. Hemidesmosomes or half-hemidesmosomes resemble desmosomes morphologically and in connecting to intermediate filaments, and like desmosomes, they act as rivets to distribute tensile or shearing forces through an epithelium. Instead of joining adjacent epithelial cells, however, hemidesmosomes connect the basal surface of an epithelial cell to the underlying basal lamina. The extracellular domains of the integrins that mediate the adhesion bind to a laminin protein in the basal lamina, while an intracellular domain binds via an anchor protein or plectin to keratin intermediate filaments. So this figure showing here is the desmosomes and hemidesmosomes. Its distribution in epithelial cells of the small intestine. The keratin intermediate filaments, networks of adjacent cells, are indirectly connected to one another through desmosomes, and to the basal lamina through hemidesmosomes. Although the terminology for the various anchoring junctions can be confusing, the molecular principles are relatively simple, as shown here in the figure below. So gap junctions allow small molecules to pass directly from cell to cell. So with the exception of a few terminally differentiated cells such as skeletal muscle cells and blood cells, most cells in animal tissues are in communication with their neighbors via gap junctions. So each gap junctions appears in conventional electron micrograph as a patch where the membranes of the two adjacent cells are separated by a uniform narrow gap of about 2 to 4 nanometer. So the gap is spanned by a channel forming proteins called connexins. By the way, gap junctions is a communicating cell-to-cell junctions that allows ions and small molecules to pass from the cytoplasm of one cell to the cytoplasm of the next. And the channels that they form is termed the connexons, in which they allow inorganic ions and other small water-soluble molecules to pass directly from the cytoplasm of the cell to the cytoplasm of the other, thereby coupling the cells both electrically and metabolically. So in this figure, 1914, determining the size of the gap junction channel, it shows here when a fluorescent molecules of a various sizes are injected into one of two cells coupled by gap junctions, molecules with a mass of less than 1,000 daltons can pass into the other cells, but the larger molecules cannot. So a gap junction connexon is made up of 6 transmembrane connexin subunits. Connexins are 4 pass transmembrane proteins, 6 of which assemble to form a channel or a connexon. A channel of a connexin is termed connexon. When the connexons in the plasma membranes of two cells in contact are aligned, they form a continuous aqueous channel that connects the two cell interiors. The connexons hold the interacting plasma membranes at a fixed distance apart. So, in this figure, in this letter A, it shows here a three-dimensional drawing showing the interacting plasma membranes of two adjacent cells connected by gap junctions. The opposed lipid bilayers, which is color red, are penetrated by protein assemblies called connexons, the green one, each of which is formed by 6 connexin subunits. Two connexons join across the intercellular gap to form a continuous aqueous channel connecting the two cells. In the figure B, it shows here the organization of connexins into connexons and connexons into intercellular channels. The connexons can be homomeric or heteromeric, and the intercellular channels can be homotypic or heterotypic. Gap junctions have diverse functions. So, in tissues containing electrically excitable cells, coupling via gap junction serves as an obvious purpose. Some nerve cells, for example, are electrically coupled, allowing action potentials to spread rapidly from cell to cell, without the delay that occurs at a chemical synapses. This is advantageous when speed and reliability are crucial, as in certain escape responses in fish and insects. Similarly, in vertebrates, electrical coupling through gap junctions synchronizes the contraction of both heart muscle cells and the smooth muscle cells responsible for the peristaltic movements of the intestine. Gap junctions also occur in many tissues that do not contain electrically excitable cells. In principle, the sharing of these small metabolites and ions provides a mechanism for coordinating the activities of individual cells in such tissues and for smoothing out random fluctuations in small molecules. So, in the liver, for example, the release of non-adrenaline from sympathetic nerve endings in response to a fall in blood glucose levels stimulates hepatocytes to increase glycogen breakdown and release glucose into the blood. Not all the hepatocytes are innervated by sympathetic nerves. However, by means of gap junctions that connect hepatocytes, the signal is transmitted from the innervated hepatocytes to the non-innervated ones. The normal development of ovarian follicles also depend on gap junction mediated communication, in this case between the oocyte and the surrounding granulosa cells. A mutation in the gene that encodes the connexin that normally couples these two cell types causes infertility, as we can see in this figure 1917, gap junction coupling in the ovarian follicle. So the oocyte is surrounded by a thick layer of extracellular matrix called the zona pellucida. The surrounding granulosa cells are coupled to each other by gap junctions formed by connexin 43 or CX43. In addition, the granulosa cells extend processes through the zona pellucida and make gap junctions with the oocyte. These gap junctions contain a different connexin. So the connexin is CX37. Mutations in the gene encoding CX37 cause infertility by disrupting the development of both the granulosa cells and the oocyte. Cell coupling via gap junctions also seems to be important in embryogenesis. The permeability of gap junctions can be regulated. Like conventional ion channels, individual gap junctions channels do not remain continuously open. Instead, they flip between open and closed states. Moreover, the permeability of gap junctions is rapidly and reversely reduced by experimental manipulations that decrease the cytosolic pH or increase the cytosolic concentration of free calcium ions to very high levels. Thus, gap junctions channels are dynamic structures that can undergo irreversible conformational changes that closes the channel in response to changes in the cell. The purpose of pH regulation of gap junction permeability is unknown. In one case, however, the purpose of calcium ion control seems clear. When a cell is damaged, its plasma membrane can become leaky. Ions present at high concentration in the extracellular fluid such as calcium and sodium and then move into the cell and valuable metabolites leak out. If the cell were to remain coupled to its healthy neighbors, these two would suffer a dangerous disturbance of their internal chemistry, but the large influx of calcium into the damaged cell causes its gap junction channels to close immediately, effectively isolating the cell and preventing the damage from spreading to other cells. Gap junction communication can also be regulated by extracellular signals. The neurotransmitter dopamine, for example, reduces gap junction communication between a class of neurons in the retina in response to an increase in light intensity. This reduction in gap junction permeability helps the retina switch from using rod photoreceptor, which are good detectors of low light, to cone photoreceptors, which detect color and fine detail in bright light. So in this figure 1918, the regulation of gap junction coupling by a neurotransmitter. In figure A, We have here a neuron in a rabbit retina. It was injected with a dye lucifer yellow, which passes readily through gap junctions and labels other neurons of the same type that are connected to the injected cell by gap junctions. In letter B, we have here a retina that was first treated with neurotransmitter dopamine before the neuron was injected with dye. As can be seen, the dopamine treatment greatly decreased the permeability of the gap junctions. Dopamine acts by increasing intercellular cyclic AMP levels. In this figure below, figure 1919, it shows the summary of the various types of junctions formed by vertebrate cells in an epithelium. In plants, plasmodesmata perform many of the same functions as gap junctions. So, the tissue of a plant are organized in different principles from those of an animal. This is because plant cells are imprisoned within rigid cell walls composed of an extracellular matrix rich in cellulose and other polysaccharides, as we discuss later. The cell walls of adjacent cells are firmly cemented to those of their neighbors, which eliminates the need for anchoring junctions to hold the cells in place. But a need for a direct cell-to-cell communication remains. Thus, plant cells have only one class of intercellular junctions. We have the plasmodesmata, or in singular, we have the term is plasmodesma. Like gap junctions, they directly connect the cytoplasm of adjacent cells. In plants, however, the cell wall between a typical pair of adjacent cells is at least 0.1 micrometer thick, and so a structure is very different from a gap junction. Plasmodesmata has a few specialized exceptions. Every living cell in a higher plant is connected to its living neighbors by these structures, which form fine cytoplasmic channels through the intervening cell walls. As we can see in this figure 1920, it shows here the plasmodesmata. In letter A, it shows here the cytoplasmic channels of plasmodesmata pierce the plant cell wall and connect all cells in a plant together. In B, each plasmodesmata is lined with plasma membrane that is common to two connected cells. This usually also contains a fine tubular structure, the desmotubule, which is derived from the smooth endoplasmic reticulum. Running through the center of the channel in most plasmodesmata is narrower cylindrical structure that is desmotubule, which is continuous with the elements of the smooth endoplasmic reticulum in each of the connected cells. So in this figure 1921, various views, it shows the various views of the plasmodesmata. In letter A, we have here an electron micrograph of a longitudinal section of a plasmodesmata from a water fern. The plasma membrane lines the pore and is continuous from one cell to the next. In letter B, it shows here a similar plasmodesmata seen in a cross-section. In C, we have a small pit fields of plasmodesmata in a cell wall isolated from a tobacco leaf. So let's proceed to the cell adhesion. What is cell adhesion? Cell adhesion is the process by which cells form contacts with each other or with their substratum through specialized protein complexes. So, to form an anchoring junction, cells must first adhere. A bulky cytoskeletal apparatus must then be assembled around the molecules that directly mediate the adhesion. The result is a well-defined structure, a desmosome, a hemidesmosome, a focal adhesion, or an adherence junctions that is easily identified in the electron microscope. Indeed, electron microscopy provided the basis for the original classification of cell junctions. In the early stages of cell junction development, however, before the cytoskeletal apparatus has assembled, cells often adhere to one another without clearly displaying these characteristic structures. In the electron microscope, one may simply see two plasma membranes separated by a small gap. Animal cells can assemble into tissues either in place or after they migrate. Many simple tissues, including most epithelial tissues, derive from precursor cells whose progeny are prevented from wandering away by being attached to the extracellular matrix or to other cells. As we can see here in figure 1922, it shows here the progeny of the founder cell that are retained in the epithelium by basal lamina and by cell-to-cell adhesion mechanisms, including the formation of the intercellular junction. Selective adhesion is even more essential for the development of tissues that have more complex origins involving cell migration. In these tissues, one population of cells invades another and assembles with it, and perhaps with other migrant cells, to form an orderly structure. In vertebrate embryos, for example, cells from the neural crest break away from the epithelial neural tube, of which they are initially a part, and migrate along specific paths to many other regions. They are assembled with other cells and with another to differentiate into a variety of tissues, including those of the peripheral. So in this figure, 1923, it shows here an example of a more complex mechanism by which cells assemble to form a tissue. Some cells are initially part of the epithelial neural tube, alter their adhesive properties, and disengage from the epithelium to form the neural crest on the upper surface of the neural tube. The cells then migrate away and form a variety of cell types and tissues throughout the embryo. Here, they are shown assembling and differentiating to form collections of nerve cells, which is called the ganglia, in the peripheral nervous system. On other neural crest cells differentiate in the ganglion to become supporting or satellite cell, which surrounds the neurons. The crest cells tend to migrate in clusters, and they proliferate rapidly as they migrate. Cell motility and cell adhesion combine to bring about these kinds of morphogenetic events. The process requires some mechanism for directing the cells to their final destination. This may involve chemotaxis or chemorepulsion, the secretion of a soluble chemical that attracts or repels migrating cells, respectively, or pathway guidance, the laying down of adhesive or repellent molecules in the extracellular matrix or on cell surfaces to guide the migrating cells along the right paths. Then, once a migrating cell has reached its destination, it must recognize and join other cells of the appropriate type to assemble into a tissue. So, dissociated vertebrate cells can reassemble into organized tissues through selective cell-to-cell adhesion. Unlike adult vertebrate tissues, which are difficult to dissociate, embryonic vertebrate tissues are easily dissociated. This is usually done by treating the tissue with low concentrations of a proteolytic enzyme such as trypsin, sometimes combined with the removal of extracellular calcium and magnesium with a divalent cation chelator such as EDTA. These reagents disrupt the protein-protein interactions that hold cells together. So cells adhere to each other and to the extracellular matrix through cell surface proteins called the cell adhesion molecules. as this comes is a category that includes the transmembrane adhesion proteins. So CAMs can be cell-to-cell adhesion molecules or cell matrix adhesion molecules. Some CAMs are calcium dependent, whereas others are calcium independent. The calcium dependent CAMs or cell adhesion molecules can be seen primarily responsible for the tissue-specific cell-to-cell adhesion seen in early vertebrate embryos. Explaining why these cells can be aggregated with calcium chelating agents. CAMs were also initially identified by making antibodies against cell surface molecules and then testing the antibodies for their ability to inhibit cell-to-cell adhesion in a test tube. Cadherins also mediate calcium-dependent cell-to-cell adhesion. So the cadherins are the major CAMs responsible for the calcium-dependent cell-to-cell adhesion in invertebrate tissues. The first three cadherins that were discovered were named according to the main tissues in which they were found. We have A cadherin, which is present in many types of epithelial cells, N cadherin or nerve cell, on nerve, muscle, and lens cells, and P cadherin on cells in the placenta and epidermis. All are also found in various other tissues. N cadherin, for example, is expressed in fibroblasts, and A cadherin is expressed in parts of the brain. This and other classical cadherins are related in sequence throughout their extracellular and intracellular domains. There are also a large number of non-classical cadherins, with more than 50 expressed in the brain alone. The non-classical cadherins include proteins with known non-classical cadherins I was wrong with the typing. So, non-classical cadherins include proteins with known adhesive function, such as desmosomal cadherins and the diverse protocadherins, which is found in the brain. They also include protein that appear to have non-adhesive functions, such as T cadherin, which lacks a transmembrane domain, and a glycosylphosphatidylinositol. Glycosylphosphatidylinositol or GPI, in which anchor, and the fat protein which anchor the nerve and muscle cells, and the fat protein which was first identified as the product of tumor suppressor gene in Drosophila. Together, the classical and the non-classical cadherin proteins constitute the cadherin superfamily. So in this table 19.3, it shows the some members of the cadherin superfamily. We have A cadherin, N cadherin, P cadherin, VE cadherin in the classical, and in the non-classical we have desmocholine, desmoglein, T cadherin, and protocadherins. Also, cadherins are expressed in both invertebrates and vertebrates. Virtually all vertebrate cells seem to express one or more cadherins according to the cell type. They are the main adhesion molecules holding cells together in early embryonic tissues. So in culture, the removal of extracellular calcium or treatment with anti-cadherin antibodies disrupts embryonic tissues, and if cadherin-mediated tension is left intact, antibodies against other adhesion molecules have little effect. So most cadherins are single-pass transmembrane glycoproteins, which is about 700 to 750 amino acids long. So we have here figure 19.24 shows the structure in the functions of cadherents. In letter A, we have the classical cadherent molecule. The protein is a homodimer with the extracellular part of each polypeptide folded into 5 cadherent repeats. There are calcium binding sites between each pair of repeats. In letter B, we have the crystal structure of a single cadherent repeat. It resembles an immunoglobulin domain. And in letter C, we have the influence of extracellular calcium. As the amount of calcium ions increases, the cellular parts of the cadherent chains becomes more rigid. When enough calcium is bound, the cadherent dimer extends from the surface where it can bind to a cadherent dimer on a neighboring cell. If calcium is removed, the extracellular part of the protein becomes floppy and is degraded by proteolytic enzymes. Cadherents also have crucial roles in development. So, a cadherent is the best characterized cadherent. It is usually concentrated in adherence junctions in more mature epithelial cells, where it helps connect the cortical actin cytoskeletons of the cell it holds together. So, a cadherent is also the first cadherent expressed during mammalian development. It helps cause compaction, an important morphological change that occurs at the 8th cell stage of mouse embryo development. So, cadherents, or during compaction, the loosely attached cells called blastomeres become tightly packed together and joined by intercellular junctions. So, cadherents are also crucial in later stages of vertebrate development. So, we have here a figure in 1925 showing the distribution of E cadherent and N cadherent in the developing nervous system. So, immunofluorescence micrographs of a cross-section of a chick embryo showing the developing neural tube labeled with antibodies against letter A, we have E cadherent, and letter B, we have N cadherent. Note that the overlying ectoderm cells expresses only E cadherent, while the cells in the neural tube have lost E cadherent and have acquired N cadherent. So, cadherents also mediate cell-to-cell adhesion by a homophilic mechanism. So, how do cell-to-cell adhesion molecules such as cadherents bind cells together? There are 3 possibilities that are. The first one is in homophilic binding, molecules on one cell bind to other molecules of the same kind on adjacent cells. And the second one, in heterophilic binding, the molecules on one cell bind to molecules of a different kind on adjacent cells. And the third one, in linker-dependent binding, cell surface receptors on adjacent cells are linked to one another by secreted multivalent linker molecules. Although all 3 mechanisms have found to operate in animals, cadherents usually link cells by the homophilic mechanism. In a line of cultured fibroblasts called L cells, for example, the cells neither express cadherents nor adhere to one another. When L cells are transfected with DNA encoding E cadherent, the transfected cells become adherent to one another by calcium-dependent mechanism, and the cell adhesion is inhibited by anti-E cadherent antibodies. Since cadherent proteins can bind directly to one another and the transfected cells do not bind to untransfected L cells, one can conclude that E cadherent binds cells together through the interaction of 2 E cadherent molecules on different cells. So we have here a figure, 1926, showing the 3 mechanisms by which cell surface molecules can mediate cell-to-cell adhesion. So although all of these mechanisms can operate in animals, the one that depends on extracellular linker molecules seem to be the least common. So if L cells expressing different coherents are mixed together, they sort out and aggregate separately, indicating that different coherents preferentially bind to their own type, mimicking what happens when cells derived from tissues that express different coherents are mixed together, as shown in figure 1927. The coherent dependent cell sorting. So cells in culture can sort themselves out according to the type and level of coherence they express. This can be visualized by labeling different population of cells with dyes of different colors. So in letter A, we have cells expressing N coherent, sort out from cells expressing E coherent, which is color blue. In letter B, we have cells expressing high levels of E coherent, sort out from cells expressing low levels of E coherent. So coherents also are linked to the actin cytoskeleton by catenins. Most coherents, including all classical and some non-classical ones, functions as transmembrane adhesion proteins that indirectly link the actin cytoskeletons of the cells they join together. This arrangement occurs in adherence junctions. The highly conserved cytoplasmic tail of these coherents interact indirectly with actin filaments by means of a group of intracellular anchor proteins we called catenins. So this interaction is essential for the efficient cell-to-cell adhesion, as classical coherents that lack their cytoplasmic domain cannot hold cells strongly together. So we have here figure 99 showing the adherence junctions. So in letter A, we have the adherence junctions in the form of adhesion belts between epithelial cells and the small intestine. The belt-like junctions encircles each of the interacting cells. Its most obvious feature is the contractile bundle of actin filaments running along the cytoplasmic surface of the junctional plasma membrane. So in letter B, we have the molecules that form an adherence junction. The actin filaments are joined from cell to cell by transmembrane adhesion proteins called coherents. So the coherents forms homodimers in the plasma membrane of each interacting cell. The extracellular domain of the coherent dimer binds to the extracellular domain of the identical coherent dimer on the adjacent cell. The intracellular tails of the coherents bind to anchor proteins that tie them to the actin filaments. These anchor proteins include alpha-catenin, beta-catenin, gamma-catenin, or also called plakoglobin, alpha-actinin, and vinculin. Some cells also can regulate the adhesive activity of their coherents. This regulation may be important for the cellular rearrangements that occur within epithelia when these cells sheets change their shape and organization during animal development. So the molecular basis of this regulation is uncertain but may involve the phosphorylation of anchor proteins attached to the cytoplasmic tail of the coherent. Also, in this figure, 1929, balikan natin, it is the linkage of classical coherents to actin filaments. The coherents are coupled indirectly to actin filaments by the anchor proteins alpha-catenin and beta-catenin. A third intracellular protein called also binds to the cadherin cytoplasmic tail and regulates cadherin function. Selectins also mediate transient cell-to-cell adhesions in the bloodstream. White blood cells lead a nomadic life, moving to and fro between the bloodstream and the tissues, and this necessitates special adhesive properties. So these properties depends on selectins. Selectins are cell surface carbohydrate binding proteins or lectins that mediate a variety of transient calcium-dependent cell-to-cell adhesion interactions in the bloodstream. There are also three types of L-selectin on white blood cells. We have P-selectin on blood platelets and on endothelial cells have been locally activated by the inflammatory response, and E-selectin on activated endothelial cells. Each selectin is transmembrane protein with a highly conserved lectin domain that binds to a specific oligosaccharide on another cell. So selectins have an important role in binding white blood cells to endothelial cells lining blood vessels, thereby enabling the blood cells to migrate out of the bloodstream into the tissue. Also, selectins do not act alone, however, they collaborate with integrins which strengthen the binding of the cells to the endothelium. So in this figure, it shows the structure and function of selectins. So in letter A, we have the structure of a P-selectin. The selectin attaches to the actin cytoskeleton through anchor proteins that are still poorly characterized. So in letter B, it shows how selectins and integrins mediate the cell-to-cell adhesion required for a white blood cell to migrate out of the bloodstream into a tissue. So, members of the immunoglobulin superfamily of proteins mediate calcium-independent cell-to-cell adhesion. So, cadherin, selectins, and integrins all depend on the extracellular calcium or magnesium ions for some integrins to function in cell adhesion. The molecules responsible for calcium-independent cell-to-cell adhesion belong mainly to the large and ancient immunoglobulin superfamily of proteins. These proteins contain one or more immunoglobulin-like domains that are characteristic of antibody molecules. One of the best studied examples is the neural cell adhesion molecule, which is expressed by a variety of cell types, including the most nerve cells. So, neural cell adhesion molecule or NCAM is the most prevalent of the calcium-independent cell-to-cell adhesion molecules in vertebrates. And like adherence, it is thought to bind cells together by a homophilic mechanisms. Some immunoglobulin like cell-to-cell adhesion proteins use a heterophilic mechanism. So intercellular adhesion molecules or ECAMs on endothelial cells, for example, bind to integrins on blood cells when blood cells migrate out of the bloodstream. So there are at least 20 forms of NCAM. It is all generated by alternative splicing of an RNA transcript produced from a single gene. So in this figure, the cell adhesion protein NCAM. So in letter A, we have the four forms of NCAM. The extracellular part of the polypeptide chain in each case is folded into 5 immunoglobulin-like domains and or 2 other domains called fibronectin type 3 repeats. Disulfide bonds, which is color red in this figure, connect the ends of each loop that forms an immunoglobulin-like domain. So in letter B, we have a model for the homophilic interaction that allows INCOM to mediate cell-to-cell adhesion. So multiple types of cell surface molecules act in parallel to mediate selective cell-to-cell adhesion. So a single type of cell utilizes multiple molecular mechanisms in adhering to other cells. Some of these mechanisms involved organized cell junctions, while others do not. So each cell in a multicellular animal contains an assortment of cell surface receptors that enables the cell to respond specifically to the complementary set of soluble extracellular signal molecules. So in this figure, 1932, it shows the summary of the junctional and non-junctional adhesive mechanisms used by animals in binding to one another and to the extracellular matrix. So the junctional mechanisms are shown in epithelial cells, while the non-junctional mechanisms are shown in non-epithelial cells. So a junctional mechanism is operationally defined as one that can be seen as a specialized region of contact by conventional or freeze fracture electron microscopy. A non-junctional adhesion shows no such obvious specialized structure. Note that the integrins and the cadherins are involved in both non-junctional and junctional cell-to-cell cadherins and cell matrix or integrins contacts. So cadherins generally mediate homophilic interactions, whereas integrins mediate heterophilic interactions. The cadherins, integrins, and selectins act as transmembrane adhesion molecules and depend on extracellular divalent cations to function. On blood cells, selectins and integrins can also act as heterophilic cell-to-cell adhesion molecules. The selectins bind to carbohydrate while the cell-binding integrins bind to the members of the immunoglobulin superfamily. And lastly, we have the non-junctional contacts may initiate cell-to-cell adhesions that junctional contacts then orient and stabilize. So the interacting plasma membranes are simply seen to come close together and run parallel, separated by a space of 10 to 20 nanometer. This type of non-junctional contact may be optimal for cell locomotion, close enough to provide traction and allow transmembrane adhesion proteins to interact, but not so tight or so solidly anchored to the cytoskeleton as to immobilize the cell. So a reasonable hypothesis is that non-junctional cell-to-cell adhesion proteins initiate cell-to-cell adhesions, which are then oriented and stabilized by the assembly of a full-blown intercellular junction. So many of the transmembrane proteins involved can diffuse in the plane of the plasma membrane, and in this or other ways can be recruited to sites of cell-to-cell and cell matrix contact, enabling non-junctional adhesions to enlarge and mature into junctional adhesions. And these are my references. Thank you all for bearing with me and